Recording this question. All right.
Yep, this meeting will be recorded as it was on the email, and I just started recording it. You all must have got notified.
All right. And just let us know whenever you want us to go.
Yes, let's start. Anna, please go ahead.
Alrighty. Okay, so welcome everyone. I'm Anna Borg, mom to Sienna, a rare girl who has CDKL5 deficiency disorder, and I'm also the director of patient advocacy and partnerships at Hope for Harper CDKL5, a USA-based patient advocacy group. We want to thank everyone for joining today. Hope for Harper CDKL5 and Longboard Pharma for an update about the Pacific Research Study, a clinical study for patients with developmental and epileptic encephalopathies. Please welcome Chad Orovilo, EVP of Clinical Operations, and Dewey McLean, VP of Medical Affairs.
Great.
Welcome, everyone.
Yeah, thanks for the introduction, and thanks to you, Anna, and to Janu and Andreas and everyone that's helped us get to this point to be able to talk with you. I thought today maybe Chad and I will just introduce ourselves real quickly, and we'll just jump right into the drug we're working on and the study and what it's going to look like. So my name is Dewey McLean. I'm the head of medical affairs for Longboard. I'm a neuroscientist in my past life, and now I help companies that are working in the rare CNS drug development space as we make this transition from doing drug discovery to moving drugs forward into patients through clinical development. And Chad, you want to introduce yourself?
Yeah, hello everyone. My name is Chad Orvillo. I'm the head of clinical operations at Longboard Pharmaceuticals and one of the co-founders. So I have over about 30 years of drug development experience and excited to be speaking with you all today. Thanks so much. Do.
Great. All right. Yeah, if we can just go to the first slide. So I just want to take one slide to tell you a little bit about us, because most of you probably aren't familiar with our company. So our company is Longboard Pharmaceuticals. We're headquartered in San Diego in the La Jolla area. We're a biotech company. We're 33 employees as of now. We went public 2 years ago, almost exactly 2 years ago, actually, in March of 21. That number is a little misleading though, because we're really like a 20-year-old, 2-year-old company, because we're a spin out of a company that's been around for a long time called Arena Pharmaceuticals. And Arena Pharmaceuticals was really, led by a group of chemists who were really excellent at doing G-coupled protein receptor research and really dialing in and dialing out specific targets for drugs at G-coupled protein receptors. So our vision as a company, when they spun us out, was a world where devastating neurological conditions are no longer devastating. This is a company that had a lot of drugs in development. In fact, they've been bought by Pfizer, in the meantime, but they had several CNS drugs for rare diseases and wanted to really give those drugs the attention they deserved. So they spun out Longboard. Chad was one of the founders that came over from Arena and helped build us into an organization that can do CNS, really drug development. So, one of the things that's great about being here, I've been on board for about a year, is that the whole company from the beginning has worked really closely with the advocacy groups, including you all, trying to get input from the early stage to make sure that we're doing drug development in the right way from as early as phase 1. And really that we're looking at not just what endpoints are necessary for drug approval and regulatory endpoints that are the necessary endpoints, but also what matters to you all as families and to the patients, and in terms of quality of life and other outcomes for patients. And so I just want to say thanks at the beginning for being really good partners and working on this. None of us can do drug development without people that are willing to be involved in studies and learn about studies and be involved in research, and whether it's clinical trial research like ours, or it's something like a patient registry that's tracking the natural progression of the disease over time. Being involved in research in any way is incredibly helpful across the community to all of us. So thanks very much. All right, let's go on to the next slide. So you don't have to read all the words on the slide. I just want to bring to you a couple of the take-home points, which is our drug is LP352. It doesn't have a catchy name yet. We hope that gets remedied sometime soon, but for now it's LP352. And this drug is a serotonin receptor agonist, and very specifically, it hits the serotonin receptors that are called 5-HT2C receptors. So these 2C receptors are on the GABAergic neurons in the brain, and GABAergic neurons increase inhibition. So they are basically like brakes on activity in the brain. And so by hitting these 2C receptors, we think the mechanism of this drug is to increase inhibition and thereby tone down the hyperexcitability that happens that leads to seizures, and that leads to that epileptiform activity. And there's a lot of evidence that goes into supporting this. There's data from looking at neurons in slices. There are data from animal models, fish and mice and rat. And there's data from some other drugs that work potentially using this mechanism as well. So there's really converging evidence suggesting that if you activate 2C receptors, you turn up the inhibition and thereby decrease excitability in the brain. So that's the proposed mechanism here. If you can go on to the next slide. So I'm not going to show you all the studies because there's a host of them, but here's just an example of the zebrafish model. That's a classic one used for some developmental and epileptic encephalopathies. And on the left in the dark gray is what the regular zebrafish looks like. The green bar is the epileptiform activity that you see in these fish if you genetically modify them to be predisposed to having seizures with the SCN mutation. And then if you add our drug, you can see it decreases that epilepsy-like activity back down to normal levels. So this is one example of some of the animal data, but as we said, there's kind of converging evidence that this is how such drugs probably work. All right, let's go to the next slide. One of the questions we got, and this is a little more technical than a slide I would usually show for this kind of presentation, but one of the questions we got ahead of time was, what does it mean to be a superagonist? Because when we talk about LP352, we talk about it as a superagonist. So remember I said this drug activates the serotonin 2C receptors. So if you look here on this figure, that green line is the response that these cells with serotonin receptors have when you put serotonin on them. If you put another drug, lorcaserin, that hits those 2C receptors, you can see its response in purple, which is a drug response that activates them, but not quite as much as the natural chemical in the brain, which is the green line, the serotonin. But then if you give our drug, LP352, that's the blue line, and you can see it activates these cells even more than the brain's own serotonin does. And so we say a superagonist, that's what we mean. It means the drug is even more potent than the natural chemical in the body is, which we think, hopefully will translate into some positive efficacy as we look at it more in these studies. All right, I'm going to the next slide. So, what we're looking at here is this activity at the 2C receptor I talked about, and that's great. One of the challenges with looking at these serotonin agonists is there are a couple of different receptors. It's not just the 2C receptor. There's also 2B receptors and 2A receptors. And, unfortunately, when you activate those receptors, they can lead to some side effects that aren't really desirable. The 2B receptors lead to, or can lead to at high doses over a long period of time, valvular heart disease and pulmonary hypertension. The 2A receptors, a lot of psychedelic drugs work by activating the 2A receptor. So you get hallucinations and insomnia and other CNS effects. So one of the great things about the development of this drug is it's very, very specific for the 2C receptor. You can see here, if you look at 2C versus 2B, 200 times more selective. The same with 2C versus 2A. So you never know if you actually have the drug in people what the outcome is, but our hope is that this leads to the benefits of 2C activity with none of the liability or adverse events from hitting the 2B, or 2A receptors. All right, go on to the next slide. You know what, let's skip this one and just jump right ahead. This is just more about the selectivity. So where are we now with development? So what's happened so far is we've done studies in healthy volunteers. So there's two kinds of studies, SAD and MAD studies. It's a terrible name, but that's what they're always called in drug development. The SAD studies are the single ascending dose, and the MAD studies are multiple ascending dose. So first you do the SAD study when people get single doses of the drug, and this is really a safety study to see how it behaves and what the safety looks like in healthy volunteers. And so we've done that study up to 24 milligrams. And what we saw was very promising, which was not many AEs. The adverse events we saw were mild to moderate. And they're the things that you expect when you're affecting serotonin in the brain. So some headache, some dizziness, and some nausea were really the most common things that we saw. So after you get done with those single dose studies, then you go to another group of healthy volunteers and you give them multiple doses at higher and higher doses, again, just to look at safety. And what we saw here was also very promising. It looked very similar to what we saw in the single dose studies. Really mostly central nervous system side effects, like the headache and dizziness and sleepiness, mostly mild to moderate. And in this case, we actually did see one severe side effect, which was interesting because it was dizziness, and it was in a patient that happened 2 days after they took their last dose of the drug. So they weren't on the drug anymore when they had their serious adverse event. And, looking at this, we think was probably underlying that is you can have withdrawal from too much serotonin. And on the last day of the study, when patients were done, we just abruptly stopped them on that last dose. So to make sure this is less likely to happen in the future, now we've added a titration in. So when Chad talks about the study, you'll see instead of just starting and stopping on one day, then we'll take a few days to ramp up the dose and a few days to ramp down the dose at the end. But otherwise, mild to moderate CNS effects of these healthy volunteers. All right. So I'm just going to sum up what I have to say with this slide here, which is, kind of why we're excited about the drug and like where we see the opportunity. Again, it's really specific for this 5-HT2C receptor and doesn't hit the 2B or the 2A receptors that we know other drugs like fenfluramine or some of the psychiatric drugs hit. We know the preclinical data support this being anti-epileptic in those models, in many of them. And we know what the data look like in the healthy volunteers in terms of safety. And that's promising enough for us to move it forward into this first study in patients. to Chad. At that point, I'll hand it off to you to talk about the Pacific study.
Great. Thanks so much, Dewey. Yes, so we're excited to talk to you about the Pacific study. As Dewey mentioned, this is our first study in patients. This study is, again, looking at safety, tolerability. It looks at efficacy in terms of the ability of the drug to reduce seizures, and also pharmacokinetics. Again, it's looking at, what are the drug levels in the body as we go through this study. This is unique in that, again, it's looking at not only a single developmental and epileptic encephalopathy, but various developmental and epileptic encephalopathies, or DEEs. It is double-blind and placebo-controlled, which is typical in drug development, and that's needed because you need to really be able to assess the efficacy of your drug relative to variances in the disease over time. While that is true, that it is, again, double-blind and placebo-controlled, you have a very good chance of receiving the active treatment, which is LP352. It's a 4-to-1 randomization of active to placebo, so you have an 80% chance of receiving drug. We started this study in early of 2022 in March. We're targeting about 35 sites in the US and Australia. Most of those sites are in the US. There's only 5 sites in Australia. The medication is taken orally, and patients can have a feeding tube. Overall duration of participation is about 22 weeks. One bonus is that if you haven't already had genetic testing, there is optional genetic testing that is offered. And so that is an upside to participating in this study, along with the, again, close treatment and follow-up that you receive when you participate in a clinical trial. Patients who complete the Pacific study are also eligible for an open label extension, which takes the treatment out within the open label extension for a period of 1 year after completing Pacific. So the Pacific study is looking for patients 12 to 65 years old. As I mentioned, we're looking across all manners of DEEs. Typically, you'll be studying patients, let's say in Dravet syndrome or LGS, or one DEE in particular, but we are open to all DEEs, including CDKL5, as highlighted here, and others such as TSC, and we'll talk about that a little bit more in some subsequent slides. In order to qualify for the study, you must have uncontrolled seizures, so you must have a minimum of 4 motor seizures per month for the 3 months prior to screening. You must also be on stable doses of other anti-seizure medications. You're allowed to be on up to 4 additional anti-seizure medications, and that doesn't include other treatments such as VNS or a ketogenic diet. Key exclusion criteria. We're not allowing patients who are concurrently using fenfluramine or Fintepla or lorcaserin, which is another 5-H. These are both 5-HT2C agonists. They also hit on other subtypes, which are 5-HT2A and 2B, which you saw in an earlier study. And the reason that we're not allowing them to be in the study is because, again, it's the same mechanism of action as LP352. The other key exclusion criteria is really any history of cardiovascular or cerebrovascular disease. The Pacific Research Study website is highlighted here, but we'll talk about that more as well. So we can go to the next slide. So the study takes place across 5 phases. Initially, you'll be entering a screening period of up to 5 weeks. And really what's important during the screening period is you're completing a seizure diary to ensure, again, that you can qualify for the study based on the minimum number of seizures. It also ensures that, again, you can remain on a stable other anti-seizure medications during that time period. Once you continue to qualify for the study, you'll be randomized into the study, and this is where you'll begin to receive treatment. And this is where you won't know if it's active or placebo. Again, you have an 80% chance of receiving active medication. As Dewey mentioned, we have what's called an up titration, where you start at a dose of 6 milligrams of LP352. And as long as you could continue to tolerate it, you'll begin to increase your doses from 6 to 9 and then up to 12 milligrams. If for any reason you step up in a dose, but you can't tolerate it, you are allowed to step back down into the previous dose. Patients must be able to take at least a minimum of 6 milligrams in order to qualify and continue in the study. And then you'll enter what's called the maintenance period, where you're going to continue on that dose that you're able to up titrate to for a period of 60 days or 2 months. Thereafter, if you're not continuing into the open label extension, you'll down titrate off the medication. As Dewey mentioned, we want to ensure that patients are safe and they don't experience any of those adverse effects that we saw in the healthy volunteer study. So they will down titrate completely off. If you roll over into the open label extension, you'll down titrate to what's the equivalent of a 6 milligram dose. Again, because this part of the study is double blind, and so we need to maintain the blind. And once patients roll over into the open label extension, they'll once again begin that up titration to whatever they can tolerate into the open label extension. In terms of the numbers, we're looking for a total of 50 patients in this study. As I mentioned, we're looking again at safety. We're looking at efficacy as well. There are a couple of other outcome measures. So we're looking at what's called the Columbia Suicide Severity Rating Scale, as well as the Patient Health Questionnaire. And again, we continue to look at seizure counts via the electronic diary that also has a paper backup. Okay, next slide. As we mentioned, typically when you're studying patients with DEEs, you're going to study one particular condition. That could be Dravet, CDKL5, or LGS. This study is unique in that we're taking all comers across all the variety of the developmental and epileptic encephalopathies. So we're looking for patients with Dravet, we're looking for patients with LGS, and then we're also looking for all of the other DEEs, including CDKL5. We came up with, worked with the Epilepsy Study Consortium to define the inclusion criteria here. And what you could see here is that patients with other DEEs must have a seizure onset prior to age 5, must have a combination of various different seizure types. You must have some delayed developmental history, as well as abnormal EEGs that are showing up as slow or disorganized. Next slide. This again just further highlights the fact that patients with CDKL5 deficiency disorder, as well as TSC and other DEEs qualify for our study.
And in fact, if I can just add one thing, Chad, like, you know, if people can have a genetic confirmation of the diagnosis, or they could just have a clinical diagnosis without the genetic confirmation. And both of those are totally fine in terms of coming into our study, as long as you meet the other study seizure criteria.
Yeah, thanks, Dewey. And again, as we mentioned, if you don't already have the genetic testing, you can receive that as part of this study. Next slide. I think it's also worth saying, you know, for this study, we're providing as much support as we can. We provide support for travel as well as overnight stays. As I mentioned, we are conducting this study in up to 35, approximately 35 study centers. If you go to clinicaltrials.gov, we try to maintain the active study centers there on clinicaltrials.gov. But if you go to the Pacific Study website, which the website is given here again, it also, there's a questionnaire that allows you to see if you can qualify for the study. And that also allows you to be connected with the closest possible study site to your area. We were, again, proud to be working on the Pacific study. We believe it's a unique design in that it's a basket study, again, including all manners of DEEs, not just one specific DEE. As Dewey mentioned, we're very happy to be working with all patient advocacy groups. It's one thing that we started with very early on in the inception of the company. We believe it's very important to be connected so that we could hear you in terms of what's important, and also, you know, making sure that we can answer any questions that you may have about the study. Okay, next slide, please. Yeah, so I guess to close, again, please, if there's any questions, feel free to go to the Pacific Research Study Trial.com, the website. Again, there's a questionnaire there. There's additional information that you can receive about the study. And, you know, we'd be happy to answer any questions that you may have.
Yeah, thanks, Chad. And I'll just say, as we open it up for questions here as well, that we're really particularly excited about the basket approach here. And there have been a lot of conditions. Fortunately, in the CDKL5 space, with what Marinus has done, you guys have had opportunities to begin research in some cases, but for many of the DEEs, patients have never had an opportunity to be involved in research at all. And we really want to open it up and make sure CDKL5 and then all of the EEs have research opportunity to be involved in studies here. So I will leave it there and hand it back to Anna for questions.
Thank you, guys. This is amazing stuff. Really appreciate having prepared this. And I know it's Sunday, but yeah, we do have a few follow-up questions from parents and caregivers that were shared with us. The first question is, when and based on what factors would this drug be considered for patients younger than 12 years of age?
It's a great question. So typically, in phase 3, you will study these conditions, all the way down to age 2 or even down to age 1. That's the goal. Right now, again, part of this study is including patients that are adolescent patients. We also have a sister study that's looking. We talked a little bit about pharmacokinetics, and we have a sister study that's looking more closely at the pharmacokinetics or drug exposure in the smaller stature patients and including the adolescents. That's going to help us understand drug exposure for the younger patients. And our intent and our hope is that in phase 3, we can include, again, the pediatric patient population down to age 2 or potentially down to age 1. That's going to be a discussion that we're going to have with the agency, the FDA, in terms of our study designs to ensure that we could, again, allow patients down to that age range.
But we feel you. We have a sense of hope and urgency as well around getting down to that lowest age possible. And there are limitations on us in terms of drug development of needing to start with healthy volunteers first and then going to this first group of patients. I think everyone felt very comfortable down to age 12 and then wanting to get these data to make us feel really comfortable to go down to age 2 or 1 in the next set of studies. But our intention certainly for phase 3 is to be down to age 2 or 1 for those studies.
Yeah, that gives me hope. Thank you. All right, second question. Along with seizures, what other features of developmental and epileptic encephalopathies is the drug looking at?
Do you want me to take this one, Chad?
Yeah, good.
Yeah, sure. So, this comes back a little bit to the drug development process that I was just talking about. And so for this study, the Pacific study, really the primary outcome is safety. And we're looking at seizure counts in terms of countable seizures in these patients for this study as the first step in looking at that. Where that question is going to be really important is in the phase 3 studies. And for me, I have a bias about this, and that I've worked in the epilepsy space for a number of years, and I think seizure reduction is great, and I think we have to show seizure reduction for the FDA to approve the drug. I don't think it's what people primarily care about or exclusively care about, right? So we're still in the process of designing what those outcome measures are going to look like for that phase 3 study that comes next. But I think it has to, part of the conversation has to be communication. It has to be mobility. It has to be quality of life. And we need to work with you all as well to make sure, in addition to what we just have to do to get over the hurdle of getting the drug approved, that we're measuring things that matter. And that's certainly, why we're talking with you guys now.
And to that end, we would love, feedback in terms of what is important, what is, what else can we be looking at or thinking about in terms of our study designs in the future, so that we can help, again, design our studies, in the future for what's meaningful for all of you.
Thank you.
Yeah.
Absolutely. Okay,
It's
third
interesting,
question.
just like it was the smallest side, it's like, drug development here, it's frustratingly slow in some ways, but then in some ways in this space in particular, it's really fast because in other disease states that aren't rare disease, you go through phase 1, then you do 2, and then you do 3. Here we're doing like 1, 2 combined, and we're going to be able presumably to jump from this straight to phase 3, and so we're going to be able to jump right to answering those questions, hopefully with the next round.
Yeah, that's amazing. Alrighty, third question. How are you monitoring the effects on the heart during the clinical trial?
Sure. You'll probably notice we didn't call out echocardiograms, and I think people are familiar with Fintepla, where they have an ongoing program that monitors, again, safety via echocardiograms. Because we're only primarily interacting with the 5-HT2C receptor, not 5-HT2B, that is really the receptor that drives the cardiovascular effects, we're not required to monitor echocardiograms in our study. We are monitoring via lab results, cardiac troponins, to look at some again, the cardiovascular effects, as well as ECGs, which are typical in this stage and also future stages of drug development.
Thank you. We have one more question that just came in today. Your drug shows more absorption than natural serotonin in slide 5. Can there be side effects of too much serotonin?
Yeah, I mean, I think one of the things, and we saw it potentially in that multiple ascending dose study, is there's, for example, potential withdrawal from having too much serotonin. Another thing that people have seen with we haven't seen it with our drug ever, but with serotonin reuptake inhibitors that are commonly used to treat depression, people develop serotonin syndrome sometimes. That's one of the reasons that you don't combine this drug. So patients, for example, cannot be on SSRIs if they're in the study, because we don't want to, run any risk. Although it's incredibly rare, we haven't seen it. We don't want to see it. So, but yeah, there certainly are potential risks for too much serotonin.
I see. Yeah, that makes sense. And about drug interactions, do you guys have data on that? Any specific interactions for like common seizure medications or?
So we have an ongoing study that's looking at drug-drug interactions specifically. And again, what we're avoiding are drugs with similar mechanisms, as Dewey mentioned, SSRIs, and also, again, we mentioned Fintepla and lorcaserin. We have also in the study itself modified various exclusion criteria as we've learned more about the drug. So, for example, different anti-seizure medications are metabolized via different pathways, and the CYPs are one good example. We're not primarily metabolized via that pathway. We are metabolized via what's called glucuronidation. And so there's certain medications that we know that we don't believe we have to worry about from a drug-drug interaction perspective. So stiripentol is a good example where, with Fintepla, they know that they have an interaction with stiripentol, so they have to adjust their dose. We don't have to make any dose adjustments at this point. So,
thank you. When do you anticipate data results from the study to be available?
So we are hoping to have results the second half of this year. So that's why we're working hard to again recruit the study, complete the study, and hopefully have a data readout by the end of this year.
I see. And sorry if you answered that already, but what seizure reduction results is there so far? If you guys can share some.
Yeah. Oh, I'm sorry, Chad. Do you want to go?
No, so I mean, from the clinical study, we do not have any data thus far, so it's ongoing. Sorry, do we go ahead?
No, that's what I was going to say. Is the data that we have in hand are we've looked at those healthy volunteer studies to look at safety. We've done some other stuff that I didn't mention, which was looking at the levels in cerebral spinal fluid versus the levels in blood, and we see that it's getting from blood into the brain like very rapidly, and like the exposure in the brain looks very much like it does in blood. But this study that we're enrolling right now will be the first one where we are able to generate some data in seizure reduction. So sign up for the study, and you can be the first to know.
Yeah, no, we're excited for sure. Yeah, I don't know, Janu, are there more questions? I think we've asked all of them, and you guys just went right for them. Really appreciate that. I think this is really it. Let me just double check if we have anything else in the Q&A.
Thanks, Anna. While you double check, I wanted to take this opportunity to thank Chad and Dewey for taking the time. I understand, this is Sunday, and we just went through the time zone change, so everything is up in the air. One question that comes up is explaining the difference between the existing medicine for this, phenfluramine, and what you are doing. I know you covered this, and we've spoken before, so you did explain that in detail as well. I just wanted to ensure that, if there is anything you want to add to that and explain that to everyone, that would be helpful.
Yeah, no, I mean, so Fintepla or fenfluramine has now been approved for both Dravet and for LGS, and I think it's, on the one hand, it's a really nice proof of concept, I think, and one thing we're really happy about is seeing the efficacy that drug has, because I think it gives us confidence that using the similar mechanism of 5-HT2C, if that's really what's driving the efficacy, then we anticipate good things for the drug that we're working on, knock on wood. And I think the great specificity we have, and the hope that we have, is by having that specificity for the 2C receptor and avoiding the 2B receptor that's associated with the cardiovascular liabilities, that if the drug works and it's approved, we hopefully will not have to have echocardiograms as part of starting and monitoring and after you're off the drug. One of the other limitations, I think with Fintepla, is that there's a maximum dose that patients can achieve, and so it's not clear if going higher might be beneficial. And so we're also hopeful that not having that maximum dose might give us opportunity to optimize the dose of LP352 a little more, because theoretically won't have that limitation. So that's kind of where it is. They're in the same family, working on certainly at least both of them working on the 2C receptor, and then these differences that we're hopeful will play out into a drug that has optimized efficacy and removes some of the hurdles of the REMS program.
That's great. I think, Anna, we covered all the questions, correct?
Yes, we have, and
Okay.
This will be recorded. I mean, it is being recorded. We'll share with everyone, and also the folks who couldn't participate. And if there are more questions, we can definitely just send them to Dewey and Chad as well, and just share more information about the study and also the websites and so on. So yeah, anyone who has more questions, please feel free to reach out to us, and we'll just make sure that we get them answered.
Great.
Thanks so much for everyone's time today. Really appreciate the chance to come talk to you all. And we're very available, so we'll be looking for those questions if they come through.
Yes.
Okay.
Yeah.
Thank you so
Thank
much.
you all.
Everyone, we appreciate you. Thank you. Have a
Thanks.
nice
Enjoy.
rest of Sunday.
You too.
Bye.
Bye.
Thank you.
Bye-bye. Thank you.
Bye, everyone. Bye.